Hi, my name is Dan Carlson, and I'm the Chief Scientific Officer of Recombinetics, where our mission is to use gene editing in large animals for a healthier people, healthier animals, and a healthier planet. So we achieve our mission through three divisions. Our first division that I'm going to talk to you about today is Surgeon, where we use engineered pig models of disease to improve therapeutic development. Second division is called Regenivita. In this division, we're using engineered pigs as an incubator to develop human cells, tissues, and organs for transplantation purposes. A third division I'm not going to talk to you about today is focused on agriculture, and this one's near and dear to my heart as a former farm kid, but that's a story for another time, and hopefully we can catch up on that later. So just a few stats about the company. We were founded in 2008, and just a couple years ago, we completed a $34 million Series A capital investment round. We have 10 scientists and 4 DVMs, which I've combined to lead to 68 patents issued and 192 pending, so a pretty significant portfolio. And fairly significant funding in the grant area, with 33 grants totaling over $20 million from NIH, DoD, USDA, and other nonprofit entities. So the platform around Surgeon is using engineered pig models of human disease, and we've been using pigs as safety models for human disease for a long time. They're a great model, but there's only so much they can do as a safety model and efficacy model from a wild type standpoint. So we use human genetics to guide us, and we identify genetic mutations that can lead to disease in humans. And we use gene editing technology to replicate those in swine. And those animals can then be used by our partners in preclinical testing that will help them gain better information earlier to choose their winning propositions. And there's a variety of different applications that these animals are used for, so they can be used for drug and device development, combination therapies, new indications or line extension for an existing product, surgical interventions, biomarker development, imaging technology development, learning and behavior, and also gene therapy applications, which I'll talk a little bit more detail of later on. We've got 12 finished models that are currently available from our pipeline, and many more that are on their way. And we also do custom collaborations to develop new lines specific to the needs of a sponsor. In our cancer franchise, we have neurofibromatosis type 1 and type 2, as well as some ovarian models. And I'll talk a little bit about some other cancer models in a bit. In addition, we have a cardiac division, which has dilated cardiomyopathy, hypertension, atherosclerosis. We have a few different animal models of that. And also we have liver and metabolic disease, alpha-1 antitrypsin deficiency, and NASH that are also in our current pipeline. So these are all made with germline gene editing, so they all have an inherited mutation that drives the development of the disease. But we've realized early on that there are some limitations to that. Basically, you only have one mutation, and you can't get a variety of different phenotypes, and the genetic drivers, like in cancer, for instance, could not be predicted, because essentially you're waiting for the natural course of cancer development. So we turn to somatic cell gene delivery to try to engineer solid tumors that really are tailored to the precise needs and genetic drivers that are required for our partners to develop their therapeutic. To do this, we combine DNA transposon systems that we can use to express oncogenes, express dominant negative tumor suppressors, or express reporter genes, so we can track development of the tumors along the way. And we combine that with site-specific nucleases like CRISPR, TALENs, and ZFNs, and use those to knock out the tumor suppressor genes. We put these all in a plasma system that we can then inject into various locations in the animal, depending on where we want the tumor. And the two that we've just begun work on are in the brain for glioblastoma modeling, and in the liver for modeling hepatocellular carcinoma. And we're really excited about some recent results that we've obtained using the system. And what I'm showing you here is the brains of 2 of the pigs that were injected. And they look very promising from the gross pathology, or the gross histology. And then with a closer look and pathologist confirmation, we found that it's a tumor. So we were very excited about that. I'm sorry they had to take a look at those gross images, but I don't know if Arnold here is much better. But this gives us really the flexibility to deliver that model in the brain for a variety of purposes, really in a short time frame compared to germline modeling. Another area that I didn't mention before is that we've worked on immunodeficient pigs that we think are going to be really valuable for IND-enabling cell and gene therapy studies. And the lines that we've developed lack both B cells, so you can see the knockouts here lack B cells compared to wild type, as well as T cells, and they lack T cells in those, and NK cells not shown here. This genetics is shown to accept xenografts very well, and some of the benefits of using a pig model here is obviously they're similar to humans and available. They're cheaper than primates to use by a fairly significant margin. Another big advantage is no need for immunosuppression. So I know of several groups that have tried to work with pig models, immunosuppressed pig models, to deliver cell and gene therapies to. And there's some leakiness associated with the immunosuppression, and it's also very costly. So it's not terribly effective as a modeling system. And we think these genetic models will really help in that area. Not only that, we can engineer these to have a specific disease or disorder on top of the immunodeficiency. So if you want to use that as an efficacy model, you'd be able to do so. The last line of animals I'm going to introduce for surgeon are these somatic cell gene editing reporter pigs. And these are part of a funded project from NIH, the Somatic Cell Genome Engineering Group. And really this group is a collection of experts from around the United States, and it's all focused on the ability to engineer or edit for gene therapy in somatic cells. And one of the challenges is scaling this up from a rodent to a large animal and obviously to humans. So our goal was to develop models that could optimize delivery and DNA repair outcomes in a large animal model. And today we've got two reporters that are in the process of development. First is the modified AI9 reporter, which is very similar to the mouse allele. And essentially this reports non-homologous end joining in the form of TD tomato expression or sodium iodine symporter, which is an in vivo imaging marker, all activated by excision of this 3X stop sequence. So this is really nice because this is very similar to the rodent AI9 allele, and anybody who's had experience with that allele in the rodents could jump directly to this model. A second one is our reporter model that incorporates the traffic light design. And the advantage of this is we can measure HDR by the repair of EGFP, in addition to measuring non-homologous end joining with mCherry and sodium iodide symporter. So how does surgeon work with customers? So we prefer a partnership model as opposed to a model of dropping the pigs off at your door and saying good luck. And so we collaborate with customers to pursue your objectives. And the first way to do that is by customizing a pig to your needs or the partner's needs. We take care of the supply end of the equation, so we make sure there's an ongoing supply of animals for scale to your needs. We assist with study design, implementation, and we also assist with partnerships with CROs like Charles River. We provide data, cells, and tissues from our surgeon archive, so we maintain a significant amount of tissue and cells from these animals, so they'll be available to you. And then we can continually optimize the models as you need. So the second half of the talk is the transition from enhancing therapeutic development to actually being the therapeutic. And in Regenivita, our goal is to supply human cells, tissues, and organs for regenerative medicine and human longevity. So one of the first phases that we're working on in Regenivita is in vivo manufacturing of living cell therapies. And this starts really the same place that most other companies start, from a human donor, and could either jump all the way to the cells by isolation of the primary cells, or stem cell engineering from that human donor, in vitro production, in vitro differentiation to these final cells. In our process, we use the pig as an intermediate, and this pig is engineered in a way that it can accept these cells, and once placed in the pig in the right location, we let nature be the guide to developing these different cell types. And what we find is that a pig opens up and unlocks the potential to develop far more of these cell types on the right, in addition to, and even more than that, that you can't actually produce in vitro. And using the pig as a manufacturing platform, there's only a handful of inputs. So we can obviously engineer our progenitor cells. Whatever cells we're putting in there can be engineered at that point to have specific features. The pigs need SPF housing, and we need specific collection and processing systems to isolate and purify the cells from the pig. And then the last thing you need is a steady supply of fig Newtons, and that keeps the pigs going. So the method that this uses, obviously starting at the stem cell, these progenitor cells are actually injected into the pig. And one of our first areas that we're going to is blood products from this. And this works best by delivering the cells actually to a fetal pig somewhere between day 40 and 45. Those cells then are those pigs then are farrowed, and the animals are used for the production, the manufacturing and collection phase, and then ultimately go back into the patients. And we're excited about this because the total addressable market in human blood products is estimated to be about 20 billion. So there's no shortage of potential there. But where we think we can have the most immediate impact in a disruptive position is a scalable manufacture of defined source off-the-shelf immunotherapy cells. In this case, we'd take stem cells like we did before and either use CD34 positive cells directly and engineer those, or make single clones of iPS cells that harbor the CAR engineering components, turn those into CD34 positive cells, and then implant them into our humanized swine incubator. We've made some pretty significant progress on the humanized swine incubator, having proven all the way back in 2015 that we could generate immunodeficient animals. And then we've been adding humanized cytokines and anti-phagocytosis agents to those animals, and every additional turn of modifications that we put in this animal takes about 6 months to turn around. And then ultimately we collect and store those cells and implant those into humans for a therapeutic. One of the nice things about this is it's versatile, and we can produce cells at a high volume. So you can imagine that a humanized pig with a variety of humanized cytokines could produce any of these lineages. But by changing the different combinations of cytokines, we could favor B cell or T cell development, or generate some cell types that are really difficult or impossible to generate in vitro, like monocytes or granulocytes. And these could be engineered in the same way to have an anti-tumor effect. We're really excited about the capacity of pigs for producing these cells. In fact, one pig, just from our estimates, could treat up to 270 patients in its lifetime without any external cell expansion. So really a significant number of cells could be produced out of just one animal. And we think about the CAR T market alone growing to 9 billion by 2027. So this is really a significant opportunity, especially when you toss in some of the other cell types that we're going to be able to produce. Phase 2 of Regenevita focuses around production of transplantable human organs. So this is the higher objective for the company, is to be able to produce whole human organs in the pig that ultimately can be transplanted back into a patient. And this really relies on our ability to make tissue or ablated animals, use blastocyst complementation to restore that organ in the pig, and transplant back to patients. And the total addressable market in this area is about 50 billion. So our roadmap to commercialization, we've made a lot of progress in the T cells, platelets, and red blood cells, and through the research phase, and moving into the early phases of generating the animals for IND, enabling studies, and then exogenic kidney and heart, the whole organ franchise is definitely in the research and development stage, but we're making a lot of great progress on human cell engraftment into the pig host. We also have one line that is involving extracellular matrix improvement that doesn't rely on actual living therapy. It's just it's a genetically modified extracellular matrix material that improves healing. Obviously, this work takes some significant investment and partnerships to make it move forward. And we are in the middle of a funding round for the parent company at this point, a small funding round, and we expect that the subdivisions will have their own funding rounds later down the road. And if you want to contact us or talk about those opportunities, I got some emails on the screen. And with that, I just say thank you for your attention. Hopefully this virtual conference is going well for you. And this is a photograph of our building and when we opened it in January 2020. And we refer to our employees here as the Recombinetics because they're really embarking on a space race to be able to produce these different types of cells, organs, and tissues in the swine host. And we're really excited about these opportunities. Thank you.